Okay, thank you very much to the organizers for giving me the opportunity to present this small talk about this group. It's together, you see it's a worldwide collaboration really. I think it's an important group, and I hope I can show you a bit why I think that is. It's not mentioned a lot, I must say. Well, they're very colorful things. They're lichens. They're all lichenized with Trypetheliales. They're actually restricted to the tropics, very much so that there's not a single one here in Europe, for instance. And they're not in, well, they're a bit in USA, but only in Florida, that areas. So mostly rainforest, and they're gall-forming. I mean, mostly lichens are thought of as being quite harmless, but these are the exceptions to that rule, really. At the moment, there are only 300 species known, and most of them are not in use, in a sense. I mean, they have been described some time, and that's the only time they've ever been mentioned. That's for about half of the current species the case. And there are in 12 genera, and these genera are still, well, almost Sacchardian, I would say, Saccardian. Unlike other Trypetheliales, they're usually quite big and colorful. So they're really, you can think of areas the size of a meter square or something like that. And in the tropics, it's about the second most diverse lichen group after the Graphidaceae. And it means that, they're not on every tree, but they're almost on every tree. So if you go there, you can collect, well, in a day you can collect many specimens, and you can see in the field, that helps. You can see morphological differences. I mean, that's with the small parenocarps, that's often quite different, but these are big enough. And if you think that they are on almost all trees there, and they cover several meters of this tree because they're on the branches, they're on the bark, and this tree is quite high, and you think how much of the tropics is actually tree covered, you realize that the total surface covered by these Trypetheliales is millions of square kilometers, really. I mean, that's much more than fruiting bodies of most of the groups we are talking about. Well, this is how they look like. So I'm looking at one here, and on the other place, everything what looks a bit bulbous, what looks a bit warty, these are Trypetheliales. So there are 3-4 species on the left hand, and on the right hand there are several, but there's especially one big thallus going all around the bark, showing that it came there when the tree was still young. So it started and was a long time ago. So they're all lichenized, as far as we know. Crustose thallus. I mean, some lichens have these foliose and fruticose forms, but Trypetheliales don't have them. And ascomycetes are usually immersed, somewhat aggregated. Ascospores, I'll show some about the ascospores. The Hamatecium filaments, there's no variation in that, except that sometimes they're with oil bodies or not. There is a little bit difference between the species from exposed trees, so like these, they're often more colorful, more aggregated as ascomycetes. From open areas, they are more flush, let's say. So these are more of the common things you would see in every plantation, basically. But there are special species on rainforest tree trunks, and there even, well, the one upper left is almost squamulose. I mean, it's really, this thallus has some, is not so flat anymore. And the one below that, well, you actually don't see anything there. I mean, it's just bark. But there's a Trypetheliaceae. It's immersed in the bark, and it grows in close connection with the bark, and it often makes the bark responding to that. Here, these are the ones from the canopy branches. Originally, it was thought that these were the same things as from the open areas, but it's not. There are many specialized species just up in the canopy branches. This is the upper two ones are sections to the same species, and it shows the impact actually the lichen has on the bark, because the colored parts, so the stained parts, are bark elements. And the same with this thallus lobe there. That's one of these more or less lobed ones. And you see there's a lot of interaction with the bark going on. The bark is actually making galls in response to the lichen growing there. It's not really harmful to most of the trees, although in the middle of a big thallus, it cracks open, and then there's a potential risk to the trees. Well, here's some other structures, like they have sometimes yellow oil in the spores or yellow other things. So there's a lot with color happening. So there's lots of chemicals. Here are some of the ascospores. So most of them are distal-septate, I would say, and they all start with just the simple structure to the left. And the one to the left, you can also see that this one has a gelatinous area around it, gelatinous gel, and that can have different morphs. But you can only observe them on fresh material, of course. And looking at ascospores is still one of the things we do to actually figure out species. These are 150 specimens that look really the same from the outside, so there's no differences in the ascomer structure or in the thallus, except that half of them are containing lichenized, so that's one character. So half of these three of the species have lichenized, and the others not. And then with this character and the length and width of the spores, you can see that there are groups. It's not just one continuum. And this, I must say that all these specimens are from one locality, so from one forest, from 10 or 15 different trees, but one forest. So there's really one population, and you can figure out whether this is a species or not. Something we routinely still do is making TLC plates. I'll just show a few. There's lots of spots there, and for some we have an idea what it actually is. Often anthracenones, sometimes other quinones. Some spots you don't know. Terpenoids are turning up. I mean, you never be sure whether they really belong to the lichen. But the colored ones are certainly characters. And there's a whole diversity there, and you can use them to see whether, well, they are species characters at least. I mean, for the rest we don't use them, but the species character is good. They're pretty little studied. I mean, there's only 24 publications really, so nobody else ever did. But 3 monographs, but they're a bit old, and they had only 10 to 30 species, so there's still need for an update. Until the 50s, there were as many names as there were specimens, more or less, because every collected specimen happened to be recombined in another genus, and in another genus, and in another genus again. So many of them have 4 or 5 names, but there's still only one specimen now. So we decided to do a small monographic project, and for this we studied 800 specimens in the field. One of the main reasons that we started doing this was that there was a demand for names by ecological students who, if you go out in the tropics and want to make ecological studies, you come across this. And the Graphidacea you can now do, identify a bit, but the Trypetheliales is really difficult. So we ended up looking at a lot of types too, and herbarium specimens to make it more complete. And 200 specimens have been sequenced. Well, this is how it looks. That's the collecting we did ourselves. So on this tree, it's just a simple mango tree, and this red part is one of these red lichens, this Marcellaria was collecting. But on this mango tree, there were 12 different Trypetheliaceae, for instance. And this is a pretty good tree because it was close to the rainforest, so it will catch also capture some species from the rainforest. It's not that every mango tree has 12 species. And of course, if there's a tree fallen down, that's always a reason to stop. Well, these are these ecological students from different places, from Mexico and Brazil, for instance. The historical work is, this is what you get from herbarium in Australia. I was very happy to get this because it's on one sheet, but you're also kind of obliged to look at the other things then. And while you have it, nobody else can look for any of these things. But it usually turns out that these specimens, these types, haven't been restudied a lot. So some have been restudied once since they were described, but most have never been restudied. So some of them are not even in the family, in the genus. This is the first DNA work which was published. It was in Taxon last year. The work was done by Matt Nelson, and at the moment we are looking at a much bigger tree with more entries in it, twice as many, but the groups are still the same. And luckily some of these groups are exactly what we hoped would come out as group. I mean, there are groups that are nicely recognized by some features. For instance, if you look at Trypetheliales and Aptrootia, that's the only ones in the core which have brown ascospores. All the others have hyaline ascospores. So that's a good one. But you see from some of the other characters which were traditionally used to distinguish the genera, so for instance Astrothelium, the one on the top, and Trypethelium, they were distinguished on different ostial positions, so combined like this or vertical like that in the pseudostroma. Now that's in the characters to the left, and it doesn't fit. But there's another character that actually still makes it easy to recognize what is a Astrothelium and what is a Trypethelium, and that's the ascospore. So there's more words actually in the ascospores. The ascospore of Trypethelium have roundish lumina, whereas these of Astrothelium have these diamond-shaped lumina, which I showed you before. Well, down to the bottom of the cladogram, it's all a bit uncertain. That we are trying to add more and more, especially in that area. You don't have to read the names, but it gives an idea that the groups are really stable. Well, so far we have 400 species now, so 100 new will be described, well, next year or so, in 16 genera. And because of the generic division is now different, even if you have a big group, let's say for Astrothelium, you still need about 150 new combinations really. And I don't think anybody is going to complain about that because these things are not used a lot. And the names that are used most are actually used in a sense which is much too wide. It doesn't fit the current ideas about the species. And so it says here already also ascospore septation and ascoma aggregation were not a good character. But we also from DNA work we saw that characters like these chemicals, the anthracinones, presence or absence of that, and like xanthones, for instance, are useful characters. As is, for instance, inspersion of the hymenium. So these two paraphysis types, they were really on a species level, they're of use, not on a generic level. And to somewhat surprisingly, actually, the diversity is much higher in South America than in the other tropics. I mean, Africa is a lot undercollected, but Asia is not undercollected, and there's really much fewer species than in South America. And it's like this. I mean, so all the hotspots are actually in South America, and well, you see, India, Sri Lanka is one of the better places also, and well, New Guinea, Northern Australia. This is for the whole family. This is for one of the genera, Polymeridium, and it's even more so. So in this one place, this is the Caatinga region of Brazil. So it's not the rainforest, but it's the open savanna-like forest. Now that's really the place for Polymeridium. And you see that Australia is, for instance, also relatively good for Polymeridium. But this is not an artifact of sampling, although the numbers in Africa should be a bit higher, obviously. And all numbers should be higher, because we did some prediction. I'm not going to show the model for this, but we did some prediction based on the natural vegetation in all these squares. So these are 15 by 15 degree squares. And then you see that there's still hundreds of species to be expected. And the future is, well, luckily now more and more people become interested in these things. And well, everybody who actually would study Trypetheliales would get new species immediately, I mean, on his first trip. And there are a lot of people now doing that. These are all, and this was just one of the excursions, so we are here, Robert and Marcella is here. And all the others are students or professors from Brazil in this case. And there are many people starting to work on lichens there, as there are in Thailand and some other of the, well, China is, all these BRIC countries, I would say, these are the ones where lichenology is really booming now. And they're lucky to have much to be detected also. Thank you.